Does anyone? So, well, it's finally Farzan's turn. He was a bit reluctant for some reason. And I will introduce him. Not that he really needs much introduction, but I'm sure maybe a few of the younger folks here just mainly know him from his publication. So Farzan got both his undergrad degree and his PhD in math, actually at BU. However, he did his thesis work with Nancy Coppell, so I'm figuring he was already doing theoretical neuroscience then. And then he did successive postdoc stints first with Ron Calabrese and then with Eve, where he really turned himself into an experimentalist and a real boy, a real biologist. And then he started his own lab at NJT and Rutgers, and pretty soon also recruited Jorge there. And then they both eventually got me to join, and Farzan and I essentially run our labs together with a lot of overlap with Jorge too. He had left Eve's lab a few years, I think, before I started there. But I think I met him in my first week in the US when we all schlepped down to Woods Hole for the East Coast NervNet meeting. And back in the day, at least as East Coast folks met each other at least twice a year at NervNet and SFN. And at these meetings, I thought he was always like the guy with the smart and the tough questions, which I really liked. And I also, to this day, really like that when he's around, I'm not necessarily the grumpiest guy in the room. I guess dependent on how much coffee he had. Give me 1 second here. We then sort of got to really know each other when he invited me to help teach in the NSMB Neurosystem Behavior course for a few years. And I really learned a lot from him. He has this real no-nonsense attitude about science, a dislike of the politics that is involved there, a conceptual rigor that I really admire very much. But I think what I always liked most or admired most about Farzan is that he's not a computational guy who does a bit of experimental work, or an experimental guy who does a bit of theory, or he's also neither a techniques guy nor someone who just sort of takes established approaches. He's pretty damn good at all of these things. So he's probably one of the people with the most well-balanced skill set that I know. And I started collaborating with him because I wanted someone who can cover sort of the theoretical side of a couple of things I did. And it just turned into this truly synergistic collaboration where most of the time it'd be impossible to disentangle whose idea something was, no matter if it's experimental or theoretical approaches or whatever. So in his own work, he did a lot of things that were important contributions to our understanding of the role of things like synaptic dynamics and the role that plays for central pattern generation and neuromodulatory effects. Worked on intrinsic dynamics and resonance, and I think today he'll tell you mostly about newer stuff about neuromodulation and co-modulation from sort of cellular to circuit levels. So with that, take it away, Farzan.
Well, thank you for that generous introduction. And I am the jack of all trades, master of none. I'm gonna start by doing something so I don't have to tell you about circuit dynamics. Instead, I'm gonna tell you a little bit about my mentors and my collaborative projects. And I wanna do some shout outs here. First to Ron Calabrese, who I consider one of my main mentors. He's the person who really taught me neurobiology, and also the person who convinced me, not directly, but by his actions, that I really want to be an educator. And second, to Mike Nussbaum, who I met really and got to know. I mean, I met him earlier when I was in Ron's lab, but got to know him also at NSNB. And he taught me a lot about the details of the system, and also how attention to detail is really important, and being rigorous in your work is really important. And not the least, Eve, who has been really truly my scientific and educational mentor, and again, all three of my mentors have taught me that it's not just about the science, but it's also about giving it to the next generation and trying to train them to do things right. I also want to include among my mentors Yair, who was my I would say he was my scientific soulmate for many years. We started in Eve's lab and we were joined at the hips, really. And I'm still in touch with him. We're still friends, even though he moved away from academia and went into other. And I think he just got a master's degree in art history. He eventually went to his original love and is going to do art history. And Jorge, who I've had the pleasure of sharing my lab with for more than 20 years, it's been a real blast, and I have learned so much from Jorge that I can't even mention how much I appreciate things that I've learned from him. And finally, Dirk himself, who as he said, really I'm doing everything with him. All our projects are together, a lot of them together with Jorge, but with Dirk, everything I do is done with him. And I have also greatly come to appreciate his conceptual clarity in looking at science. I've done a lot of projects. I started my lab by doing synaptic dynamics and continue to do this. We've done this for more than 20 years now. And there's some things that we've learned from this that I find quite invaluable, and a lot of collaborations, including with Bruce, who I see here. We did a couple of papers on that, and with Ron Harris-Warwick. And inter-circuit interactions, the gastropyloric interactions, the many papers we did with Mike Nussbaum, starting from when I was in Eve's lab and together with Yair and Marlena Bartos, and going on to other sets of projects. Unfortunately, I never got funded on this, despite the many papers that came out, but it was a blast working on it. The electrical coupling project, mostly a modeling project that I did a lot with Jorge. It was something that became a kind of a pet theoretical thing that Jorge and I did. And Jorge talked about this in his talk, so I'm not going to go in detail into this. I have done a fair number of math and computational modeling work and papers, including with Eve after Cristina joined my lab for a while, Cristina Soto Trevino, and also with many of my collaborators at NJIT. And then I got into this kind of black hole of looking at membrane potential resonance, something I'm still somewhat involved in and collaborate with Horacio Rothstein, who's also next door to me right now at NJIT. And another shout out to Dirk, the role with of axons in neural coding, and we've done a whole bunch of things there. And the real shout out goes to Nelly, who's really carried the burden of doing all the experiments for this project for the past several years. And we are lagging behind in putting out the papers, but it's been a blast. And Dirk talked about this, so I'm not going to go into that. Finally, I'm going to talk about this particular topic, which is co-modulation. To get into that, I'm going to give you a little bit of background, and I'm going to go relatively fast through this because most of this is stuff you know. As with perhaps most of the people who work on the SDG, I going to focus this project on this circuit, which is the pyloric circuit. This is the Cancer Borealis version of the circuit. These are my own recordings, 4 cells and 4 extracellular recordings, the pacemaker group of AD and PD neurons, the follower neurons, LPPY, IC, and VD. And you see this beautiful triphasic rhythm, very regular when you take the prep out and put it in the dish, and quite easy to identify cells if you're doing your recordings right, and you have a nice prep. And we know that this circuit, from the work of many giants in the field, we know that the circuit is subject to modification by many chemicals that arrive as neurotransmitters or hormones that modulate the properties of neurons and synapses in the system. I just put this up to kind of show you all the different substances that we know of, and many of which that have been studied in great detail. Now, one of the things that we know about neuromodulators is that modulators shape the pyloric circuit output in terms of the pyloric circuit. So this is kind of a more historical thing. This is the good old days when you had to control pyloric rhythm in the dish from 3 extracellular recordings. You can see the triphasic rhythm. You take all of the modulatory inputs arriving to the STG out by so-called decentralization, or blocking the descending inputs through the stomatogastric nerve. And in the old days, often the rhythm basically went away. And then in the presence of any of these modulators, a variety of them, you could get some version of the rhythm back. Now, I'm gonna come back to this picture, which is a classic really in the field, although its secrets are kind of with Jim Wyman. I don't know exactly where and how he did this, but he in sequence kind of applied these different modulators to show that each of these modulators kind of carve out a different pattern of activity from the pyloric network. Now, I want to also distinguish here the actions of the excitatory neuropeptides from the actions of the amines. And I'm using the schematic that Dirk has made, which is kind of nice, that shows that if in a particular STG cell you have neuropeptide receptors, we know that these neuropeptides converge in some of their actions. In particular, they converge to activate this modulatory inward current that we've come to call IMI, whereas the different amines, in this case, dopamine, they act on a G protein coupled receptor as well, but they can act to, let's say, activate some ion channels or ionic current types and reduce the amount of other ionic current types in this cell. And in a different cell, you may have different receptors for different neuropeptides, but they still act on the same IMI, whereas this dopamine receptor may actually be doing opposite things in cell 2. So in terms of understanding how these modulators are targeting the ion channels that are their targets within the cell, and to some extent the synapses, which I'm not showing in this schematic, the actions of the peptides seem to be somewhat simpler here. Now, in terms of what this IMI, this modulatory inward current is, again, we pretty much all of us know the history of this. It's an inward current, very much like a persistent sodium current. It's a fast-acting, non-inactivating current with an IV curve that looks like an inverted bell shape. And you can see it here. And we also know from work that Andy Swenson did with Eve that even though the peptides converge in their actions to activate this current, different cell types may have different receptors for different types of peptides. Therefore, there is a different action of these peptides at the circuit level when you look at how they activate this particular current as well. Now, another issue that I wanted to mention is, and we just published this, so it's another shout out this time to Anna Schneider, but also to Jason Rodriguez, who may have characterized the first version of this, in that historically, again, IMI was measured by after blocking calcium currents, but if you don't block the calcium currents, you realize that proctolin and perhaps other peptides. Here is a ramp of voltage in the LP neuron going up from -80 to +20 millivolts and ramp back down to -80 millivolts. And this is the current that the peptide activates. And what you see is that on the ramp up, you see a bigger inward current than on the ramp down. And this size of this current, the one that activates on the upward ramp depends on the slope of the current, on how fast you go up. When you come down, the size of the one you come down does not depend on the speed or slope of the ramp. The one that you're coming down and you measure is the standard classical IMI. The one that you see on the left is the sum of IMI and the transient current, whose amplitude depends on the slope of the ramp. And if you look at the IV curve, so this is in time. Now I'm taking voltage versus current and showing how the current on the up ramp changes with the slope. You can see that it gets bigger and bigger the faster this slope is. So we call this a transient IMI. And I think in Mikey's paper with Jason and Don, they called it the transient inward current or something of the sort, or the low threshold inward transient current. Now, the interesting thing is that this current is clearly permeable to calcium. And in this particular paper of Anna's, she shows that the current, when you look at the activity of the LP neuron at different frequencies, can have very different amplitudes, which IMI in its classical form cannot reproduce. This is not what I want to talk about. This was just to mention that although I'm going to focus on IMI, there is another current that is activated potentially by other peptides, but definitely by protein. Now, the thing that I want to talk about today mainly is that modulators don't come one at a time, even though we have studied a lot of them one at a time. I just put this picture up with the schematic of a human brain and the different modulatory centers, primarily the aminergic centers, but there's a few peptides like orexin, other transmitters like acetylcholine, GABA thrown in here. And we know that even in this case, a lot of these neurotransmitters, for example, if you look here, the series of neurotransmitters that are mentioned here, act to basically do more or less the same thing or similar things on their target. This is review from David McCormick, and I don't know who the first author is actually, Zaga, but what David McCormick has shown in his work is that a lot of these modulatory inputs on thalamocortical neurons actually go in to activate the H current, basically. They converge on the same target, very similar to the peptides in our system. But effectively, a lot of these things are simply depolarizing the cell to take it from this burst mode to this tonic discharge mode. All right. And they are present together in the brain. I want to come back to the question of how do these convergent neuromodulators interact? I'll define convergent a little better in a second. So what I want to talk about is convergent of circuit output at two levels. One, I'm going to tell you stuff that is to some extent published, and that is the quantitative rules for co-modulation. And when we talk about co-modulation, we also have to be careful about the dosage of the different modulators. So this is a dose-dependent factor, and you'll see that when I show you the data. And the second thing I want to talk about is how co-modulation can influence inter-animal variability of circuit output. And it's a hypothesis that we have entertained, and I'm going to show you some data that is pretty much all unpublished at this point, although a lot of the pieces are complete. Now, we, in both cases, examined the modulators with convergent actions in the sense that they have overlapping subcellular targets, not necessarily exactly the same, but they at least overlap in some of their targets, let's say on IMI or on properties of a particular synapse, and they increase the activity. It's not that some of them increase and some of them decrease. So I'm excluding explicitly in this context the action of amines, which could be a lot more complicated if you want to look at the co-modulation effects. Specifically today, I'm going to tell you about these three modulators, Proctolin, CCAP, and RPCH, whose activity patterns I have snipped out from this figure of Jim Wyman and Eves from that paper to show you here. Now, in the question of how do different modulators act on the same target protein, on the subcellular target, now that could be, for example, an ion channel, it could be a synaptic release mechanism, or it could be a gap junction ion channel. One possibility is that the two different neuromodulators act on receptors that share somewhere downstream the same signaling pathway, in which case the actions of the modulators will occlude one another and their actions will linearly sum until you reach the point of saturation. Now, there are alternatives to this. One alternative is that there's convergence in the pathways, but they don't actually become the same pathway. They lead to different effects on the target protein, for example, by phosphorylating different sites on that protein. And in that case, you may get nonlinear effects. You may still get some sort of summation, but it doesn't have to be linear, and you won't get occlusion. You can have crosstalk in the pathway, so you can have this first case, but you may have communication in the pathway, in which case you also may get nonlinear effects. And a final possibility is that the actions are spatially segregated, in which case you do get linear summation of the actions of the modulators, but you get no occlusion of one by the other because they're acting on different targets in different parts of the cell. Of all of these, clearly, this one seems to be the simplest one, in which basically what we're saying is that the two receptors for the two modulators converge and act through the same pathway on their target. Now, if I want to understand how two modulators coming in and acting on these receptors influence this target protein, I can do a dose response curve individually for modulator one, let's say the blue one. So at any concentration, I see its effect on the target protein. I can do a dose response curve for modulator two, and I can predict that these two effects add linearly. So if I have modulator one at concentration one, modulator two at concentration two, I can add these two effects to each other. And I know that if I add these and I go beyond the level that is saturation, then I have to cut it at saturation. Okay, so that's my rule. It's not exactly linear. It would be linear if I didn't have saturation. But because I have a saturation level, it becomes this kind of linear cutoff, so it's nonlinear technically. Now, we're going to look at this in co-modulation by Proctolin and CCAP. And the first part of this story, which I'm going to tell you, which Lily did in my lab together with, in collaboration with, of course, Dirk, is looking at the effects of Proctolin and CCAP alone. So here is in her hands what Proctolin does to this particular decentralized control rhythm, what CCAP does, what they do together. When you look at the activity, this is in a single prep, you see that the phases of the activity of the LP and PD and PY neurons are different in the three cases. Okay, I'll come back to the issue of phasing later on. Right now, I'm going to look at their targets. What are the
Farzan?
targets? Go ahead.
Can I ask a quick question? I'm sorry to interrupt. I just want to ask, to what extent have you really resolved the extent of convergence? So you showed that there's this rate dependence of the current amplitude with Proctolin, right?
Yes.
And you see that regardless of what peptide you evoke IMI with. Is that correct?
The transient component of IMI, we have only done that with Proctolin. We have not looked at that in another case. The story I'm telling you right now is only about the synapses and the non-transient, the classical version of IMI, which is a very fast current and it's non-inactivating. So I don't have that particular issue.
Okay.
Okay. So we haven't gone back to look at co-modulation of the transient component. So in this case, no problem. What Lilly basically looked at the simple hypothesis that I told you, that the Proctolin receptor and the CCAP receptor downstream converge and basically influence the synapse and the level of IMI in each neuron that they're targeting by a simple summation rule up to saturation. So this schematic is the linear summation up to saturation hypothesis. Now, to look at this, she first started with the synapses. And in this case, she was recording the PD neuron with 2 electrodes, LP neuron with 2 electrodes, and she was looking in dual voltage clamp at the LP to PD synapse, which you can see here in control and in the presence of 1 micromolar Proctolin. You can see this has been published before she did this stuff, and we know that Proctolin increases the strength of the synapse and changes the activation curve of the synapse. She also did this in the opposite direction, which she called the PD to LP, and we explained that really is the summated response of the AB and PD neuron synapse to the LP neuron, in which case I'm showing you an example of what CCAP is doing to that synapse. Now, in each case, you can look at this graded synaptic current, and you can build an activation curve from your measurements. So we put voltage on the x-axis. We put the amplitude of the synapse on the y-axis. In this case, we looked at the mean amplitude during the pulse. And we look at how the modulator influences this activation curve by looking at how much it changes the amplitude, the slope, and the slope factor, the slope midpoint and the slope factor of this activation. Now, we can do this in a dose-dependent manner for each modulator, but we have a complication. So in this case, I'm looking at the LP2P synapse, looking at different doses of CCAP, and I'm going to quote Ron Calabrese on something that he said. When I say I did it, I mean we did it. When I say we did it, I mean they did it. So everything that I say we did, in fact, Lilly did. I didn't do anything other than come and tell you about it. But in this case, increasing the dose of CCAP increases the strength of the synapse, but you can measure the strength of the synapse at any voltage. So you have to kind of extrapolate and interpolate among these points to get this sigmoidal activation curve, which is the voltage dependence of the synapse, and the sigmoidal activation curve, which is the dose response curve of that synapse. So you get this surface that is kind of a double sigmoid activation. No matter, it's a very simple surface. It's just 2 sigmoids with 2 variables that are acting independent of each other. We can do this for each of the synapses, LP to PD synapse, for CCAP and for Proctolin, and for the PD to LP synapse, similarly for CCAP and Proctolin, and we have a dose response curve of each of these synapses under each condition. And now I can do the following. I can say, for example, if I go in and add Proctolin and CCAP together at different doses. So let's say I go in and put 1 nanomolar Proctolin, right, and 100 nanomolar CCAP, that would be this triangle. I can ask, how much is the synapse changing? And can I predict it from those surfaces that I gave? The way Lilly did this is that she took this combined grid of all possibilities. She divided it into 4 sets of experiments. They're the circles, triangles, the diamonds, and the stars. So in each experiment, let's say in circles, she went at 1 nanomolar CCAP, consecutively increased the level of proctolin, and in the end, saturated both. Or for the stars, she started with 100 nanomolar proctolin and 10 nanomolar CCAP, increased CCAP concentrations in the presence of 100 nanomolar proctolin, and then saturated all of them. So in each case, we know the control without modulator, and we know the total saturation with both modulators maxed out at 1 micromolar. Now, in each of these cases, what you can see, and each experiment was repeated many times. So each circle was done several times. So in this case, 1 nanomolar CCAP plus 100 nanomolar proctolin produces the dose response curve across several experiments that you see with this magenta shaped approximation and the sigmoid that's fit to it, right? What you see in the gray is that without any modulator, the synapse would have looked at the bottom here. At the very top, with both modulators saturated, the synapse would have looked like this. So in this case, this is the range it would have gone. What you see in black is the prediction that you get from linearly adding the dose response effect of 1 nanomolar CCAP to the dose response effect of 100 nanomolar proctolin. In this case, you get an almost perfect match, saying that indeed these two modulators are converging to give you linear summation. Now, what about different concentrations? Here's a bunch of other examples for this particular synapse, and you see we get very good matches. And here's the overall concentrations of proctolin and CCAP, of how well we can predict the outcome of co-modulation by knowing the dose response curve of individual modulators. In the left here, what I've done, or what she's done, is basically in all the measurements, she's put the predicted level of the synaptic current that you get, the individual dose response curves added together, cut off at saturation, versus actually measured. And overall, we get a 90% prediction of all the variability in this synapse for the LP to PD synapse. For
Farzan, I have a kind of a technical question here, and that is, if I caught you correctly, like about 10 slides ago, when you first showed us the response over time to application of a modulator, I believe you mentioned that you're averaging the entire the response over the entire time.
Correct. This is the mean response over that pulse.
Right, and I did see some dynamics there. Right, there was an upstroke and then almost like a little overshoot.
That's correct.
And so I'm wondering if these, I mean, these are amazing correlations, right? But I wonder if they would even be higher if you took the steady state response.
We haven't done the steady state. We've done the peak, and which actually in the paper, if you look in the data supplements, the peaks are also in the tables. But we do have the steady states. It's a very good question. We just haven't looked at it. Very good question. We looked only at the mean, at the peak, and they're very similar in terms of how well you can predict. The PD to LP synapse is also very good, not as good as the LP to PD, probably because by voltage clamping PD, you don't have complete voltage control over AB. So in that case, you will miss some of the control of what you're doing with the synapse. But again, it's a really amazingly good rule that these two synapses, the effects of the two modulators simply add linearly. Well, I mean, big deal. Now, one of the things that Lily noticed is that because she's voltage clamping the cells and she is stepping the voltage up over a good range, in the LP neuron, we know there are receptors for proctolin and CCAP. From the same voltage steps that she's using to measure the synaptic current, she can actually look at the intrinsic current in LP, in control and in the modulator, and calculate the level of IMI. So she looks at this current injection, she looks at the end of the 500 millisecond pulse, and at this point, whatever current is activated, inward current, we call that IMI. And here is an IV curve for this particular example I'm showing you at 0.1 micromolar proctolin.
So now you're actually doing the steady state.
Now I'm doing the steady state, but for the IMI, not for
Right.
the synapse, okay, for IMI. So here we're ignoring the peak, we're doing this. Now you can do this, of course, all the different levels of these currents, and you can fit these with kind of inverted Bell curves as you get from a simple Boltzmann equation multiplied by a driving force, which is what we did. And you can get a dose response curve for proctolin and CCAP, okay? So that's what we are measuring as a dose response curve that I'm showing you for proctolin and CCAP. The current is measured here, I think, if I remember correctly, at -10 millivolts. All right, so from these, can we predict if I, let's say, apply proctolin at 1 nanomolar and CCAP at 100 nanomolar, I should get this much IMI from CCAP, I should get this much IMI from proctolin. I add these two numbers together, I should be able to predict what this combination is with the same rule. Okay, that's what she tried to do. She looked at the prediction rule now for IMI, and this is what we got. What we have is that what we predicted is with the error bars shown in gray for all the combinations of concentrations, and what we measured is shown in pink. What I want to point out to you is, one, we are always measuring a current that is smaller than what is predicted. Two, when you get to saturating levels of proctolin and CCAP, our prediction gets better, but at lower than saturation levels, it's awful. If you put this together, this is the overall R square I get of predicted versus measured. The individual cases, I mean, this is R squared not in the sense of Pearson correlation. This is R squared as a predictive measure, so it can have negative values. If you don't believe me, look it up in Wikipedia. In this case, negative values means we did awful. 0 means that we would have done better if we picked the mean to predict, right? So we're doing really poorly, except when we get close to saturation. Well, close to saturation is just saturate, you add them, you're still saturated, your prediction is good. Okay, but below that, what we are seeing is that if I get an IMI out of, let's say, 100 nanomolar CCAP, in the presence of 1 nanomolar proctolin, I get much less IMI. And vice versa, they're inhibiting one another. I'm predicting a lot more than I'm actually getting. So we went back and we modified our model to actually put this cross inhibition in the middle there. So where proctolin and CCAP were converging to the same target, we separated their downstream target and assumed the cross inhibition there. And by doing that, assuming we have a single cross inhibition rule for all the measurements that we did, we could improve our overall R squared to 0.72, okay? If we assume individual experiments have their own amount of cross inhibition, then you can overfit the data. I confess it's overfitting, but you can do it perfect. But this is just to point out that cross inhibition is sufficient to explain what is going on. Okay, so what did I tell you so far? What I've told you is that the co-modulation of the voltage-gated current and the synaptic currents follow different rules. And surprisingly, the co-modulation of the synapses is linear, but that of the intrinsic current isn't. And the surprising part in the surprisingly is because the synapse has two sides. It has a presynaptic side and a postsynaptic side. And as we have seen, both of these can be modulated. Despite that, we get a linear summation rule. But for IMI, this work is published. So let me move on, unless there's questions on this, to the stuff that isn't. So just continuing with this theme, on the neuromodulation of components, Lilly also looked at the neuromodulation of electrical coupling conductance. In this case, she looked at PD to PD. What better two cells to pick? They're pretty much identical, or we assume they are. And the way she did it was that she voltage clamped both with two electrodes, held one at minus -60 millivolts. The other, she also held at minus -60, but then she stepped it up and down in increments up to minus -40 and down to minus -80 and measured the current in the post-junctional cell. Okay, so what you're seeing is the electrically coupled current when both cells are voltage clamped. Note that the biggest current that you get, the most positive current, corresponds to the most negative step here, which is the light blue. Because outward currents are by convention positive. So this slope of this is the electrical coupling conductance, assuming we ignore space clamp problems. Okay, so if we didn't have space clamp problems, I am not measuring, she did not measure coupling coefficient here. She measured coupling current and therefore coupling conductance directly. We've also checked with the coupling coefficient, and it gives you similar results. If you account for the amount of leak you measure in the post-junctional cell, you can get the same thing. Now, what she saw was that, so I'm only showing you the post-junctional current. The steps are exactly the same in the pre-junctional cell. And by the way, she always did it symmetrically, from one PD to the other, and then back from the other PD back to this one. So 2 points of measurement in each case. What she saw is that, interestingly, both RPCH and CCAP seem to act on this electrical coupling by increasing its conductance. And we've done this every which way. We've done it in a frequency-dependent manner, which I'm not going to show you today. The part that is really surprising is that Proctalin does exactly the opposite. So you apply Proctalin and you get a weaker gap junctional coupling between the cells. You can do this in separate experiments, or in this case, this effect washes very easily. You can do it sequentially in the same experiment, in fact. Now, since we're talking about co-modulation, what happens if you co-modulate? So in this case, she's starting at a control level and she's normalizing to that control. Proctalin is reducing the strength of the coupling, whereas CCAP is increasing it. And if you add both of them, then you get a coupling strength that is still bigger than control conditions, but is not distinct from each other. Okay, if this bigger than control conditions is not significant in this case. Similarly with Proctalin and RPCH, they go in opposite directions, but when you put them together, they come to a midpoint and they agree. If you add all three, Proctalin, CCAP, and RPCH, which she did in these set of experiments separately than these set, you also get that you don't make it any different than having two of them. And to some extent, the effect of the modulation actually washes out even when you apply all three of these. Okay, now what this means is effectively that the same neuropeptides that I was telling you have convergent actions are affecting the electrical coupling conductance in opposite directions, and the peptides that have opposite effects can cancel each other out on that coupling conductance effect. And right now, Nelly is actually doing a set of experiments trying to decide whether this has any functional consequence on the modulation of the PD-PD synapse when you actually take the PDs, and in her case, she's driving one PD at different frequencies and trying to see whether the coherence or the correlation between the activity of PDs comes apart when the coupling gets weak and becomes stronger when it doesn't. And I don't have a conclusive result to give you on that right now. This is the coupling of the components. And I apologize, I'm running a little behind, but bear with me. I'm gonna tell you a different side of this story now, none of which is published, although the electrical coupling stuff isn't published either. So far, basically, we've said that co-modulation rules must be resolved because they define different network dynamics in states of the central nervous system, and they're different rules. You can see it even in this simple case. And for the different components, and even two modulators targeting the same neuron, you can get different effects. The story I want to tell you now comes from a very simple observation that is captured here from this really nice schematic that Dirk made. How does peptidergic co-modulation affect inter-animal circuit output similarity? It's a mouthful. Across animals, the circuit outputs have a certain degree of similarity. I'm going to use this word similarity as the opposite of inter-animal variability of what Jorge has called population variability. The observation that Dirk is kind of capturing here is, suppose you have this 6 hypothetical peptides that are acting on these on the pyloric circuit 6 neuron types in the following manner. They just have receptors, these neurons, for the different peptide types. Now, if you co-apply these peptides 3 at a time, so these are different applications of 3 peptides that are shown here with these different horizontal arrows, you see a lot of cases that pretty much everybody gets modulated if you apply 3 peptides. And in some cases, it doesn't even have to be overlapping peptides. So 4, 5, and 6 can produce the same saturation modulation effect than 1, 2, and 3. So if that is the case, and we expect that by adding this excitatory input, you produce more similarity in activity of the neurons, then what we expect is that the degree of similarity should get better with co-modulation. That's the hypothesis I want to test. Now, there's several points to make here. Now, one is that we need to think about how individual modulators and single modulator influences variability across animals. I'll come back to that question in a second. We also need to think about the concentration dependent effect again, right? And in particular, does it matter that we're doing this at saturating concentrations, or do we need subsaturation levels for this? And at what level do we expect these outputs to become more similar? Now, all of this comes back to the point that we know that the intact pyloric circuit output is indeed very similar across animals, at least in the phase of activity of the neurons, if not in all aspects. But is this consistency appearing at the level of individual neurons and synapses, or do you need the whole circuit to do this? So I'm going to ignore this last point, which I call the nonlinear, the secondary effect, which is basically all of Mikey's career. I'm sorry, Mikey, which is that modulators, and maybe Don's also, modulators are not applied to a circuit by just dumping it on a circuit. Modulators come and target specific neurons, and they target it very precisely. I'm just going to ignore this at this level. I'm doing the first order approximation. I'm going to go after this question first, and this is Anna's data I'm going to show you. Do individual neuromodulators reduce variability of a single neuron output? So what she did is she synaptically isolated the LP neuron by putting the PrEP in picrotoxin and hyping both PD neurons to remove all the synaptic inputs. And then she measured a variety of responses of the LP neuron to a bunch of things. So in this case, she looked at the FI curve, measured different levels of DC current, looked at the spike frequency, fit that with a simple function. So this function has 3 parameters. I0 is the really the real base almost, right? It's a point you get spiking. A is how much spiking you get, and B is how nonlinear this curve is. So if B becomes closer and closer to 1, it becomes more and more linear. If it's closer to 0, it becomes more and more nonlinear. So I have these 3 parameters. And then she also looked at the hysteresis of this effect. If she puts pulses and increases these pulses and then decreases the pulses, she gets different levels of spike frequency. She took a range of, I think, 2 to 4 nanoamp and looked at the difference in spike frequencies on the up and down steps of FI curve. And what she found is that Proctalin or Proctalin plus CCAP applied at saturating levels both enhance the properties of the FI curve in the sense that they increase the spiking. You see A goes up, and increase the, they decrease the hysteresis level and make this curve more nonlinear. They bring it more to a saturating level curve. I'm going to ignore the I0 real base, but in all cases, they reduce the variability of these factors. So in these 2 cases, A and B, she measured variability by coefficient of variation. For A and hysteresis, which is also in Hertz, it should have been in Hertz. This is just the standard deviation over mean. For these 2 factors, because these are interval factors, she's measuring simply the standard deviation. In all these cases, the variability of the parameters goes down in the presence of a single modulator, but the second modulator, adding it, does not reduce variability. She also looked at post-inhibitory rebound properties of this cell, so just inhibit it for about 10 seconds with -5 nanoamps, hyperpolarize it, sorry, let it go, look at the latency to the first spike after you release it, and then look at the spiking as a histogram, make a cumulative histogram curve, and look at kind of when you get to the midpoint saturation, how many spikes you get for 5 seconds or 10 seconds after that, and what is the slope of this. So what is the structure of this histogram of spiking that you get upon rebound from burst, and what is the latency? And again, when you look at these parameters, they all change with a single or 2 modulators. Some change with single and not 2, and some don't change, like the slope factor. But in all cases, the variability is decreased by either single or 2 modulators, but the 2 modulators does not enhance the single modulator effect. Finally, she looked at rebound from rhythmic hyperpolarization and looked at a similar set of properties and found that, and in addition, because the LP neuron, you can see, has a slow kind of memory that Eve has published previously in a paper, that its rebound to spike changes across different pulses of inhibition. We also measured the time constant of that. And we found that, again, for all of these factors, variability is reduced with modulation, but not with dual modulation. Okay, so we can explain the reduce in variability of the FI curve and the variability in post-inhibitory rebound with computational models. The variability in the FI curve at this point, I think we can explain analytically. And it's not an effect that is due to the saturation of the response of the cell. That we have worried about this enormously. And we can discuss it later on. It's not a saturation effect. It's just a property that increasing excitability simply does this. Now, but I'm going to put the question there anyway. Is the reason that co-modulation, not modulation, not producing more reduction in variability, the fact that in the LP neuron, co-modulation simply is saturated, right? CCAP really occludes the effect of prostaglandin. It doesn't work both ways, but could it be a saturation effect? So Anna went back and did this instead of at the saturating levels of the modulator. She looked at the mid level. She actually looked at 1 nanomolar and then went to the mid level of modulation. And what she found is basically the same, except that mid level of modulation, some factors don't reduce their variability. Like in this case, hysteresis variability is not reduced. The other FI curve variability factors are reduced with the exception of the Rio-base factor, which is I0. And here she's done prostaglandin, prostaglandin CCAP, and prostaglandin CCAP and RPCH. What is constant is the sum of the level of modulation. So we're not adding modulators on top of each other. We're making sure the total modulation is at 3 × 10⁻⁸. And similarly, variability is reduced by individual modulators, but co-modulation does not reduce variability at the single cell level for rebound relationships for this periodic input for this single cell response, right? So at the single cell level, I apologize, co-modulation, even at mid levels, does not reduce variability, even though single modulation does. But does co-modulation bring the circuit output, the whole circuit, towards more similar levels, as I was telling you? And we gave this problem to Liz Cronin, who's doing her thesis with Dirk and me. And she basically went in and said, well, why don't we start with these 3 modulators and look at pairs of them and ask ourselves, is the circuit output, in these different cases becoming more similar to each other? If I put prostaglandin plus RPCH, CCAP plus RPCH, or prostaglandin plus CCAP, do these become more similar to each other? Okay. When she tried looking at this and did a bunch of experiments, she realized something interesting, which she brought to us and said, well, how different are these individual modulatory states to begin with? Is the prostaglandin output really different than CCAP output and really different than RPCH output? Do I even know this before I go to this combination? And I immediately say, yes, this is probably well known. And, falling back on what I've been pushing on you, going back to Jim Wyman's picture of this, just look at it. They're different, right? But in fact, in all of the published papers, all the differences that have been measured are between individual modulators and control conditions, but not between different individual modulators. All right? So to do that, what we've had in our mind, which I'm trying to represent schematically here, imagine this is in some high dimensional space of circuit outputs, right? You have a bunch of control inputs with modulatory inputs intact. It's pretty tight. You have a nice tight group of, circuit outputs across animals. You decentralize and you make a mess of it. Everything goes all over the place. It's big. Then you modulate this with prostaglandin, RPCH, or CCAP, and you expect it comes closer to control, comes back towards that. But each of this is kind of carving a different piece of the space, and that is its hallmark set of activities. Okay, what I'm showing in the big dots here is, let's say, the mean of the activities that we have here. And this is the whole population showing the variance too. But what if it isn't like this? What if it's like this? What if the controls are kind of a wider range, and when you decentralize, it goes all over the place. And when you modulate it, you get a ton of overlap with everything. So that means that the modulatory state, not just the modulatory state, but the control state, has a large range that may have a ton of overlap. It reminded me of what Eve said once in one of her big talks at SFN, which I took to heart. It's a beautiful quote, which is not mine, Eve's, which is, if we don't understand the limits of what is normal, how could we understand what is a disorder? And it's a similar problem. If I don't understand the limits of each of these states, then what does it mean for me to say there's a proctolin current, the proctolin circuit output? We need some sort of distance metric for these populations, which is what Liz produced. And this distance metric includes things cycle period, in fact, cycle frequency measured from the period, the phases of activity, the spiking of the different neurons, number of spikes per burst, intra-burst spike frequency. Putting this all together, looking at these different outputs of the circuit, she went in and measured this in a heroic set of experiments in different sets of animals individually modulated by either one of these. She has large numbers of these. Then she went in and said, well, I'm trying to compare apples and oranges, cycle frequency and phase and spike number and spike frequency. And we decided that one way of doing this is because we have such a large population of parameters of animals, is simply z-score all of these, right? Assume that each individual case has a distribution, subtract the mean and divide by standard deviation, and find the z-score of this 12-dimensional output, circuit output set, and then look at the distance. And this is real data. I'm only showing the centers. This is what you see in purple is the center of all the intact rhythms in the space of the first three principal components. This is the decentralized case. This is what proctolin does. It almost brings you back in center point to the intact rhythm. And this is what RPCH and CCAP do. And these are the three principal components, but I'm going to ignore that. These are the actual distances measured, intact to decentralized, decentralized to the three states. This is what's important to us. How different is proctolin from CCAP from, I have this marked wrong because, no, I have it marked right. Proctolin is distinct from both CCAP and RPCH, although more distinct from CCAP than RPCH, but CCAP and RPCH are not, in terms of these 12 parameters, distinct outputs, okay? We have only analyzed, she has only analyzed outputs that have a triphasic rhythm. If something is going, which it does sometimes with CCAP and RPCH, into these modes of activity where the LP neuron does one burst and the PD does several, she has ignored those. So qualitative differences are ignored. This is quantitative differences. Okay. We get a similar set of results from throwing all of this into a machine learning algorithm and say, we don't know anything about what you're doing, just go in and classify for us whether what we're giving you as intact is really intact, whether what we're giving you as decentralized is really decentralized, and CCAP and proctolin. And we get a very similar set of results in that intact and decentralized are easy to identify for the machine learning algorithm, but the others become a lot more ambiguous, especially between CCAP and RPCH, where the cross ambiguity is large. Okay, let me finish this story now by coming back. So I told you all of the outputs, but I'm going to keep this one hypothesis here, which Liz came up with, and I kind of buy it. We have some evidence for this. And she claims that the similarity in the peptidergic modulation of the circuit arises primarily from the peptidergic effect in exciting the pacemaker group. So long as you excite the pacemaker group, everything else becomes similar. That's how the pyloric circuit is built. Okay, that is a hypothesis that we're going back to test with dynamic clamp. But I'm pretty sure it's true from evidence we have from previous experiments, which I'm not going to tell you about. Remember I told you co-modulation does not promote single output similarity? Let me come back now to circuit output similarity. I told you the individual cells, but let me come back to the co-modulation of the circuit output similarity. And instead of going saturating levels, we're going to go at low levels of modulation, which is nanomolar, or mid levels of modulation, which is 3 times 10 to the minus 8 molar, or 30 nanomolar, which we call low or mid here. And here are the keys are either proctolin and CCAP, or proctolin, CCAP, and RPCH, and these are Anna's results. And I'm going to just show you the outputs. The cycle frequency, you look at it, with low-level modulator, the coefficient of variation gets better than decentralized. But mid-level, as you increase the number of modulators you're applying, you're making this better and better in terms of circuit similarity at cycle frequency level. How about phases? Phases in this case, she's measuring circular variance. This is shout out to Ron, who convinced me that that is the correct way of measuring variability of phase. And in this case, again, phase of PD off, LP on, LP off, PY on, PY off, all of them get better with mid-level co-modulation. Remember, this is 1 modulator, 2 modulator, 3 modulators. They all get better, but not at low level. At low level, they get somewhat better sometimes than the phase, than the variability of decentralized, but not by adding co-modulation. And again, at the spike numbers and spike frequency, with the exception of spike numbers in the PD neuron and spike frequency in the PD neuron, which the 1, 2, 3 don't change the coefficient of variation, everything else becomes more and more regular and more similar across animals as you go mid-levels of modulation. Again, total amount of the 3 modulators is the same. So it's always 3 times 10 to the minus 8. The only question is it 1, is it 2, or is it 3 at that constant total concentration? We reduce variability by increasing modulators. So co-modulation at mid-concentration levels does bring circuit output towards more similarity. We have convincing data on that. It doesn't do this at low concentrations. To understand this, we are going back to circuit models to try to put this together. Hopefully at some point I'll tell you that story. I don't have that to tell you right now. And we know at a single neuron level, this does not happen. Final slide, just bringing this back to what it means. As many of you know, there's certainly all of you who are in my institute, for the past 2 years, I've been teaching this course on the biology of addiction. And when I jumped into this and started learning about the modulatory systems that are involved in addiction, I really fell in love with dopamine. Dopamine does the most amazing things. It does all of the learning processes. It does all of the initiation of movement. And I thought this is the coolest neuromodulator ever. Until I learned about acetylcholine. So dopamine is in the midbrain. Acetylcholine has 2 primary centers in the brainstem and in the basal forebrain. And acetylcholine is amazing. It not only modulates everything that dopamine is doing. In fact, dopamine doesn't do anything until acetylcholine permits to do it. But you need it for all sorts of things. You need it to pay attention. All your attention is controlled by acetylcholine. It's the coolest modulator. Until I learned about serotonin, right? Serotonin, I won't even be conscious without it. I won't be awake without serotonin. The number of things that serotonin does in the brain is mind-boggling, right? Now, of course, these are not the only ones. Not only do we have more amines and more other types of small molecule neurotransmitters, look at the peptides we have in there. Look at just the opioid peptides that we have that are all over. They're always there. They're always acting together. I have shown you snapshot pictures. More and more, I am convinced that the snapshot pictures are just going to give us exactly that, snapshots. There's a ton of dynamics involved in when and how these things come and change things. And I think all of the rules and all of the mechanisms that I was telling you about in the SDG is just kind of cracking open a door on that circuit in what we have to do in this system. And with that, I will shut up now. And if anybody has questions, I will ask for questions. Thank you.
Yeah, I have a question, Farzan. First of all, this was amazing. Somehow it seems like you and I speak the same kind of language, so I can really follow what you're saying. But I noticed that you were using the term better when you were describing how modulation is reducing variability across the population. And I wonder if that was accidental or deliberate.
It was accidental.
Okay.
I apologize.
Because I was wondering,
is it really
better?
Subjective term on that. No, it's not better. No.
Yeah. Is it really? Yeah. Because as we all know and have discussed many times, there is a lot to be said for having variability across individuals.
No, I not only shouldn't I use the subjective term of better, but variability by itself is also, I've been so confused in trying to figure out what to do with variability. It's like, Dirk had this eureka moment the other day and jumped out at me and said, so variability has absolutely no meaning. And we've come across this many, many times. If you compare variability across different levels, there is no such thing as absolute variability. It's very subjective, it's what you say it is, right? It's only when you compare states that are comparable to each other that you can talk about variability in a meaningful way. So does the variability of X in one state change when you take the system to a different state? You're looking at the same thing, but in different states. That we can do. And hopefully that's what we're going to stick to do.
Okay.
Ron has a question.
So remind me, is there a dose response of variability across animals to a single modulator?
There is certainly a dose response of variability of animals to a single modulator. I just don't know what it is, but I know that, we have evidence of it because we've taken some snapshots of variability of properties of circuit output at different levels of modulator and co-modulation.
So then you talk about that you're always using 3 × 10⁻⁸, but you don't, that's not comparable because the potency of the different modulators that make up the 3 components varies.
Correct. Yeah.
So the total modulatory effect is going to be different across those comparisons?
The total modulatory effect is going to be different, and potentially that is why it's affecting variability that way.
Yes.
And remember, I'm picking a point 3 times 10 to the minus 8 that is smack in the dynamic range of all these modulators as far as we can see. So if you go a little lower, a little higher, you are changing the effect. Yeah, for sure.
Okay, yeah, I just wanted to clarify that.
No, you're absolutely right on that.
If I can comment on that, I mean, for all of these experiments, you kind of want to avoid that, but rigorously, you should really do every one of these experiments twice. One which keeping the overall concentration the same, and one where you do this additively, which asks slightly different questions. But that's, of course, that's getting insane pretty fast in terms of experimental matrix.
All right. Once again, apologies for going so long. I know Dirk hates me, but
Can I ask a question, Farzan?
I'm just indifferent.
Go ahead.
Thank you.
And
Very
Adi.
nice and exciting talk. Just to add complexity, does timing affect state dependence or order of application of neuromodulators?
I see
Okay.
Dirk smiling, and we have preliminary data, I should say, if not anecdotal evidence, preliminary data is almost the same, right? That yes, it does matter in some cases. Now, it gets tricky fast. Like, are these effects that you're looking at transient, and if so, how transient? So, for example, we have some evidence that potentially applying low levels of dopamine before applying a peptide has a very different result than applying the low levels of dopamine after applying the peptide on the circuit output. Now, perhaps if you keep it in that level for a few hours, it'll go back to the same thing. But it's not too surprising considering, I mean, think of what dopamine does in our brain. Dopamine's actions, there's 2 levels. There's the tonic level that is there and just enables things, but everything else is transient. It has to be transient. Timing is essential for some modulators to do their job properly, right? So it wouldn't be surprising, and it's a project that we have an army of undergrads who are doing nothing on it yet, but they are working on it. So they haven't produced any results, but we're going to look at this exact question. Does that order matter? Does the timing matter?
Right, and just to add to what you said, of course, transients could be very long, but there could be also like multi-stability, as you
absolutely.
shown in the past, and that would
for all I know, that effect that you see if you apply dopamine, and I really want to see this. I don't remember if Liz applied the dopamine at nanomolar or micromolar. I think it was micromolar level. She never went above micromolar until I screamed at her to try 10 micromolar. And she eventually did it. But it seems like the effect is just there. If you pre-apply dopamine at micromolar, put in a peptide, you just don't get the rhythm. You just don't get a consistent rhythm out of it. And put the peptide, you get a perfect rhythm. You put the dopamine on it at micromolar, doesn't do a thing.
very interesting.
It's weird, but that's what it seems to do. So
Thank you.
okay, well, thank you all for your patience. And I think our next speaker is already scheduled, is Dawn, on January 28th, right?
Yes.
Thank you all, and have a great break, great holidays, and don't get sick, please.
All right.
Thanks,
Hi, everyone.
Thank you, Farzan.
Bye.